Hai, Assalamualaikum. Welcome back from a week-long holiday. Selamat Hari Raya to those who are celebrating and to everybody else who I hope had some nice holiday, even though I'm sure most of you were also restricting yourselves to still follow some social distancing, physical distancing measures. But I hope it was an enjoyable Hari Raya anyway. So we actually just have one lecture this week, and it's actually a new lecture which I have not had a chance to cover in previous years because we never seem to have enough time, and this time we have enough time. You know, the semester has been extended, so we're going to cover a little bit more, and then after this lecture I'm not going to have a quiz for this specific topic, but we will have a test for all of the topics that have been covered in part 2, likely on Friday, so I'll update you on that, but tentatively on Friday, and I'll open it up for about a day for you to complete, and I'll give you more information about the test in one of the announcements. But basically this topic will only be included in that test and not as a separate quiz. So this topic is covering mucosal immunology. So it's a topic that's quite near and dear to my heart, very related to some of the work that I was doing in my PhD. So forgive me if I go off tangent every now and then. But basically at the end of this lecture, I hope you'll be able to explain the features and the role of mucosal immunity, especially the role played by IgA, so secretory IgA specifically. And then also be able to describe some applications in the development of mucosal vaccines. And we'll see how they're a bit different from the regular vaccines that we talked about earlier. So first, let's recap. Remember again, what is the mucosa? So the mucosa, or the mucosal membrane, is basically the epithelial tissue that lines your body's cavities and essentially forms a barrier between the external environment and your internal organs. So you've got the skin, that's the external layer for your external, and then you've got the insides that is still exposed to air and other external factors, but it's not the skin anymore. They're mucous membranes. So the mucous membrane is basically the epithelial, and then the mucus that is covering that area. So the mucus is, I'm sure you're familiar with mucus, you know, or at least what that might be like. So it's kind of like this gel fluid. It contains mainly water. It also has mucins, which gives it that kind of viscous property, some salts, proteins, lipids, and also a lot of antibodies and other types of proteins. So this picture just gives you an idea. For example, this is the intestine, and you basically have the tract, and then you've got the mucosal layer. You've got the lumen, which is where your external environment is present. So anything you eat or anything you drink that's coming from outside will first encounter this mucosal surface, which consists of your epithelium, your lamina propria, and then that's covered by connective tissue and muscle, and further wrapped as a full organ. So the mucous membranes are very important because it's present widely across your body. It lines many tracts and structures. Also in some areas, your eyelids, your trachea, your nose, your lungs, your stomach, your intestines. You've got a lot of these membranes covering those. And although it's not something that we typically consider a mucosal organ, the liver is also an organ that contains hepatocytes, which are basically modified epithelial cells, and they line the sinusoids, the cavities within the liver. So if you recall, the liver is also a place where a lot of things get metabolized, your blood cells, a lot of detoxification occurs there as well. So that also encounters a lot of things that come from external environment. And I'm a bit biased because I particularly like viruses that infect the liver, so hepatitis viruses and their role as a mucosal surface as well. But these are basically the typical mucosal organs that we think about: the lacrimal and nasal glands, the tracts. You've got the gut. The gut is a huge one when we talk about the mucosa. So the intestinal mucosa, obviously where a lot of your food gets absorbed. So also potentially where a lot of bacteria and viruses also might get entry. The stomach is a bit too harsh, because of all the acids and all that. So that's not exactly the same. And you'll notice here you've got a lot of these Peyer's patches, especially these immune structures that are only present in the intestinal mucosa. But there are similar types of immunological structures across different parts of your mucosal membranes. So this is basically what we call the mucosa-associated lymphoid tissue. So in the gut, or they'll call it the gut-associated lymphoid tissue, or if it's the nasal-associated lymphoid tissue. But they're basically present in the mucosa, and they're lymphoid tissue. So they actually play a huge role in regulating the immune defenses within these tissues. And the main features are some structural or innate barriers, and then you've got the cells there, the immune cells, the innate leukocytes present there. And for mucosal tissues, the secretory IgA plays a very important role as part of the immune response. So the innate barriers first, we don't think about it that way, but actually a lot of the microbiota or the commensal organisms that live within our tracts, they are actually the first line of defense for us, because as long as they're there, any pathogens from the outside will struggle to even find space to be able to enter. So as long as they're there, they constitute the first barrier. And then on top of that, you've also got the mucus. The mucus is also the structure that will prevent entry of any external pathogens, and the mucus is also influenced by the presence of the commensals. So they play actually quite a big role in mucosal immunity. And then the structure of the surface of the mucosa. So this is the brush border, which has the glycocalyx. So I think this picture can show you an image quite nicely. So you've got the enterocytes, you've got the microvilli, you've got the glycocalyx. On top of that, you've got the mucus layer, any unstirred water layer, and then you'll have the commensals floating somewhere here, or anchoring here, preventing the entry of any other pathogens. So these structures are basically together trying to repel any pathogens, or if they also have hydrolytic enzymes, they can also degrade any microbes or toxins that's made by the microbes. So if, let's say, there's a breach of that, or rather part of that defense is also the innate cells, the leukocytes mainly, but also the enterocytes. So that's the cells lining the gut, the epithelial cells lining the gut. So they also express pathogen recognition receptors and pathogen-associated molecular patterns that they will respond to when they're stressed, or they will just respond in a different way if they recognize it's just from commensal organisms. So this will result in some cytokine secretion, and what kind of cytokines will depend on what they're responding to. Then you also have these special cells called M cells, and the M cells, they're very large, and their role is really to sample antigen. So unlike the enterocytes, which have that microvilli and the brush border and the glycocalyx, the M cells don't have those. So because their job is to actually bring in some of the antigens, and they will transfer it to the APCs. So they will selectively sample, not just allow anything, but they're there just to make sure the leukocytes in the MALT area are aware of what's actually outside. Whereas the enterocytes and the mucus and all that, they just block everything out. The M cells will kind of monitor actually. There are a lot of things not coming in, but what's actually trying to get in. So the M cells will be able to do that. They'll pass it to the antigen presenting cells, and also be able to stimulate the lymphocytes through the APCs. So these include T cells, like the natural killer cells, natural killer T cells, the macrophages, neutrophils, dendritic cells, the memory CD4 and CD8 T cells, and memory B cells as well, and the Th17. So remember, the Th17 is a very important subset of the T lymphocytes that are present in the mucosal areas, in the gut especially. So pictorially, this what it might look like. You've got the Peyer's patch, and this is the dome area that contains a lot of the DCs and the M cells that, as you can see here, where you have the enterocytes, you've got sort of like that brush border and the glycocalyx and the commensals. But where the M cells are, you don't have all that, and the M cells is there to actually go and put out and feel and pull in some antigens to bring in to the follicle. Yeah, so that's basically how they work together. The next very important component is the secretory IgA. So if you recall, secretory IgA is an antibody, and a form of antibody. Antibodies in general are glycoproteins, and they're produced by B cells or plasma cells that's already been activated by its corresponding antigen. So there are multiple functions of antibodies depending on their type as well, but some of them are very good at fixing complement. So this will actually activate the complement system and result in lysis of cells, cause a lot of inflammation as well. They also just bind to the antigens or the microbe just to mark them better for phagocytosis, or they can also mark them for mast cells, or they can actually bind to these antigens and then also allow them to be transferred as well. So there's a lot of things that antibodies can do, and there are different types of classes as well, because their functions relate to their structure and their class. So the basic structure, if you remember, is the Y shape, and that's typical of IgG, IgD, IgE, IgA as well. But then you've got IgAs that also exist as dimers or polymers, and IgMs that exist as pentamers. So most IgMs will exist as pentamers, so very large aggregates of these monomers of IgM linked by the J chain. Similarly, the IgA dimer is also linked by a J chain. The others tend to exist as just the monomers, the Y-shaped monomers. So that's basically where we enter the talk about secretory IgA. So before secretory IgA, there's polymeric IgA. And there are actually different subclasses of IgA as well, IgA1 and then IgA2. Some different variations in their structure. And a lot of the polymeric IgA or the ones in the gut are usually of the IgA2 subtype, and a lot of the monomeric ones tend to be IgA1. But basically, anytime they dimerize or polymerize through the J chain, they're called dimeric or polymeric IgA. And then once they have secretory component on them, they are now called secretory IgA. So don't worry, I'll explain how this happens in the next few slides. But this is basically what we're talking about. The key player, mucosocytes, is this type of antibody, the secretory IgA, that is basically a polymer or a dimer. So I think this is a nice overall picture, just showing all the immune players and responses in the gut specifically. So you've got, again, the picture of that dome area with the M cell and enterocytes, brush border, commensals, the dendritic sites, the lymphocytes aggregated in the patches. And then once they actually differentiate and start maturing, they'll produce the secretory IgA, which then gets exported out into the lumen again. And this is a better look, or a closer look at these antibodies. So that the plasma cells will be then switched to produce IgA, specifically polymeric or dimeric IgA, which then binds to what is called the polymeric Ig receptor, or the polymeric immunoglobulin receptor, the PIGR. It then gets transcytosed or transported through the mucosal epithelial cell, or the enterocyte, and then at the end of it, where the site that's facing the lumen, the PIGR will get cleaved, and a portion of it, the secretory component portion, is then bound to the polymeric or dimeric IgA, and it's now called secretory IgA because it has the secretory component. So that's basically how that antibody is produced at the mucosal surface. So one thing you might want to appreciate is that in most animals, IgA is generally dimeric, or the most of the IgA made is in a dimeric or polymeric form. But in humans and in primates, about 10% of this is polymeric or dimeric, and the rest is monomeric. And they're not really associated with mucosal immunity the way the dimeric and polymeric types of antibodies are. So in the serum, you don't have a lot of this antibody subclass, or this polymeric form in the body. And this basically just shows you again what I just explained. When you have the plasma secreting IgA at the mucosal surface, it will first bind to the PIGR, the polymeric Ig receptor, on the lamina propria side. It gets transported across the cell, and then when it reaches the apical surface or the mucosal surface, then gets cleaved off, and then released as secretory IgA. And you've also got some secretory component released even without any IgA bound to it as well. So that's where you get the secretory IgA. And once it's there, it basically is the main antibody that does a lot of the work for the mucosal response. So that's basically it. So there's a few different pictures. So if you don't understand it from one picture, hopefully you'll understand it from the second or the third picture. Again, you've got the monomeric IgA, and when you've got two of them together, that's now called the dimer because it's now bound by the J chain. Now when it gets transcytosed, the mucosal site, it first binds to the secretory component of the polymeric Ig receptor, and then that blue part, the secretory component, gets cleaved off from the receptor, and then it just forms part of the secretory IgA that will be found on the mucosal surface. So the immune response favors the secretory IgA because the secretory IgA is actually less inflammatory compared to other antibody classes and antibody subtypes. So the role of secretory IgA is a few things. So it's immune exclusion, it's anti-inflammatory, and it also provides homeostasis of the commensals. So that's basically the overreaching role of secretory IgA, and most of it is because it has its structure is quite stable, even though the mucosal surface can be quite an aggressive medium with all that mucus. And so it can actually stay stable in the mucus. It has heavy glycosylation, also adds to the stability. And it's actually got a Fab independent recognition of antigens, and because it can be transcytosed and retrotranscytosed, so it's also quite useful for antigen sampling as well. So transcytosed is when it's the antibody or the dimeric or polymeric IgA transverses from the basal side to the apical side, but then let's say it's bound to some antigen. Now it can actually travel back through the enterocyte back into the lamina propria side. So quite important as well. But the main role is that it performs immune exclusion. So it limits the number of microbes that can actually access the mucosal barrier in the first place. So just by sticking to a lot of the antigens or the bacteria or viruses that enter the lumen, it then prevents it from sticking. And then when the gut, say, has peristalsis, it just gets washed out and goes further down to the large intestines and then gets processed out. And it also actually allows for the control of the commensals as well. So the secretory IgA will selectively bind certain bacteria over others, and they will suppress the expression of certain bacterial epitopes. So this will also present to certain subsets of T cells that are more tolerogenic. So secretory IgA actually plays a role in pushing the immune response to be more tolerant than inflammatory. Because if you think about it, the gut is encountering so many types of microorganisms, and a lot of them are going to be food type of antigens, or potentially not harmful bacteria in food, or just from the external environment, and you don't want to keep on having inflammation, because that will be damaging to the gut as well. So they will only switch to the more inflammatory responses if they detect that, okay, this is actually a certain pathogen that is going to be dangerous for the body. So most of the time, the secretory IgA will just coat the commensal bacteria and just keep them in the lumen, and not allow them to get close to the mucosal barrier. Only when it's necessary, then it will actually translocate. So usually that's also through the M cells. So so there you have basically the commensal bacteria, the dimeric IgA or the secretory IgA, just sticking along to the bacteria, and so with the antibody there, it can't actually adhere to the surface of the cells anymore, and so they'll just be washed out. On the same idea, secretory IgA is actually a really key component in maternal milk, because obviously that's a mucus, it's basically a modified sweat gland, right? So it's producing or excreting a lot of antibodies, and the excreted antibodies are typically secretory IgA, or it could also be IgM or IgG, but secretory IgA is the main one. And the secretory IgA plays a very important role to shape the commensal bacteria in the baby's gut as well. So that breast milk containing the mother's secretory IgA will shape the microbiota architecture, you would say, of the baby's gut, and that will then also influence, whether or not they're more likely to develop allergies, or whether they are resistant to certain types of intestinal pathogens. So it plays a huge role in the baby's gut defenses as well. Right, so understanding that, the mucosal immunity and largely the secretory IgA plays a huge role in really overall immune defenses. And I didn't include it in the slide, but they have done studies where they show that when you stimulate one mucosal site, you actually get an immune response in some other mucosites as well. So there is basically some communication or translocation of the information that certain mucosal sites receive, or any antigen stimulation that a certain mucosal site receives. So there's actually a huge potential to target mucosal sites more effectively, especially when we talk about developing vaccines. Because by and large, the vaccines that we have now are not mucosal. They're all injections, so they're all systemic. They're just given into the blood, uptaken, and then we hopefully generally stimulate the immune system. But of course, with injections, there are some problems. So a lot of the safety issues, or a lot of any adverse events, usually relate to the fact that it's being injected. So the injection site might get a rash and things like that. And issues of compliance. So compliance is also related often to this fear of needles. So people don't like to get shots because they know it's unpleasant. Children also, obviously, parents don't like bringing the kids and having the kids be injected and crying, and then they'll have irritation. So a lot of those things relate to the fact that vaccines are injected. But another really important limitation of injections is also that you need to have the injection materials. So you need to have sterile needles. You need to have somebody who's trained to be able to actually administer injection safely. So you when you want to mass immunize people, it costs a lot to have all these things in place. And on top of that, the injected vaccines typically don't actually provide protection at mucosal site. So where stimulation of a mucosal site can also result in protection in other sites, the immunization through injections doesn't mean that your nose is protected to that same antigen, for example. So that's why, like, when you get a flu shot, a flu injection, you can still get colonization in your nasal area. You still have nasal carriage, even though you technically won't get as sick from the flu because you're now immunized against the flu, but you can technically still spread the flu if you sneeze or cough and things like that. So that's why the idea of having mucosal vaccines is quite attractive, because you'd have protection from even entry for the bacteria or the virus. And a lot of bacteria and virus gain access to the body through the mucosal sites. So if you can actually use mucosal vaccination, you'll get both. You'll get ideally systemic, and also you'll get the mucosal site protection as well. But you don't actually have a lot of mucosal vaccines currently licensed. Here I put pictures of 2 that are used in humans. The FluMist isn't used very commonly anymore. They showed some data that it wasn't so effective compared to the flu shot. So the flu shot is more often used, but the FluMist was basically a mucosal vaccine because it goes through the nasal spray, so it covers your nasal surfaces and passages. As you can see, the way it's delivered doesn't require a needle. And then you've got Rotatak, which is the vaccine against rotavirus, so a very common cause of illness for babies and small children. And so these are given as oral drops. So you can see here the way it's delivered is different as well. No needles and syringes. This table gives you a list of different mucosal vaccines that are licensed. Not a lot of them are for humans. Quite a few of them are for animals. So you've got different types of delivery routes: oral, intranasal. Basically, these are the 2 main ones. It's either oral, intranasal. Hypothetically, that you can use other routes of entry, but these are probably the 2 easiest ones to target for. So some challenges in making mucosal vaccines. One, obviously, you've got a lot of routes to choose from. So you need to choose carefully, because not all of them will give you the same type of response. It'll depend on the organization of that route, of the lymphoid structures present in that route, and also what your target organism is. So, for example, would it make sense if you want to immunize against flu by giving an oral drop? That's not where the flu is going to enter usually. Not that it might not be present. Like, for example, with the coronavirus, we know that it's actually able to target multiple sites, but the first point of entry is the respiratory system. So it makes more sense to give you a mucosal vaccine through that route. But, with delivery, eating things is always easier. So that might be something to consider as well. But another challenge is that with mucosal immunity, as we just learned with secretory IgA, it tends to be biased towards more tolerant responses. They don't want to respond through inflammation if possible. They want to tolerate whatever they encounter. So those are things that need to be considered, like you might need to use adjuvants and things like that. And also the other consideration would be the size of the vaccine, because the size of the vaccine determines whether or not your target route of entry can even uptake the vaccine. So, for example, the goblet cells in the passageways can allow entry of soluble protein antigens, but not inert particles. And then there's a specific size range that they won't accept. Enterocytes will only endocytose soluble particles 20 to 40 nanometers. It's fairly small. M cells for larger ones above 100 nanometers. And then also the size of the vaccine will determine how the immune system responds, so and where it gets delivered. So anything less than 5 micrometers will be delivered to the MALT, and then anything larger than 10 micrometers will be taken up by alveolar macrophages and dendritic cells. And it also will shift whether or not it's a T cell response or a B cell response, so whether it's cell-mediated immunity or humoral immunity. And this picture basically shows you that the larger the particle, the more likely that it will induce an antibody response, and the smaller the particle, the more likely it will induce a small a cytotoxic T cell response. And I mean, this may also relate to whether or not it's able to be endocytosed or processed as an extracellular or intracellular type of antigen. So there's some developments on this front. So mucosal vaccines are very attractive, and there's a lot of work being done here. A very interesting idea is the idea of using plants as bioreactors to make vaccines. So obviously, if you make an edible plant and they have vaccines, you can just eat it, and then you would be vaccinated through the gut. But you could also take the plant extracts, turn it into drops, same idea. And then there's also some other ideas like the immunostimulatory complex technology that's been used already for veterinary vaccines. And this all relates to better delivery of the vaccines, because when you're introducing it through different routes, you need to think about how you're going to protect, preserve the antigen integrity, increase the bioavailability. That means how much you give is how much is absorbed and taken up. And then also whether or not that influences how the immune system processes the antigens. So there are things people investigate, like using polyethylene glycol, PEG, in nanoparticles. So this has been shown to be effective if you want to deliver hepatitis B antigens in mice through the oral route. Chitosan is another type of substance that's biodegradable, biocompatible, non-toxic, and they've used that for E. coli vaccine formulations aimed for oral delivery as well. And then some other substances like alginate and other nanoparticles that just basically wants to increase viscosity and increase retention time. So if you recall as well, like a lot of times with the mucosal surface, there's a lot of peristalsis at the gut, for example, and the secretory IgA is out there just to stick onto things and then let it get washed away. So they have to do certain things to the vaccine so that it gets retained a bit longer and has a chance to actually be uptaken. So there are other carriers that are being looked at for mucosal vaccines, like liposomes. So these are basically vesicles that are composed of lipid bilayers. And they can also use virus-like particles. So these are basically protein structures that look like viruses, but they don't have the genetic material of viruses. So they just contain the antigens. And another very interesting one, particularly my interest, is using a bacterial vesicle. So bacteria naturally produce vesicles for different reasons: communication, to stimulate the immune system in a certain way. So they will also contain a lot of antigenic, immunogenic proteins and glycoproteins, and these can be harvested and then used as potential vaccine candidates as well. The only problem is they might also be toxin-like, or they'll have toxicity. So they would need to be treated in a certain way to reduce some of that toxic capacity. But also, often if you do that, you might lose that immunogenicity as well. They usually come hand in hand. But it's an interesting idea. So this picture shows you gram-negative bacteria and the vesicles that they make. They're called outer membrane vesicles. And gram-positive bacteria also make these type of vesicles, and they're called extracellular vesicles or membrane vesicles. And they contain a variety of antigens, and even sometimes some nucleic acids, RNAs, and things like that. A lot of lipoproteins as well, usually. So these are very interesting candidates as well that people are trying to investigate as new vaccines, especially for mucosal vaccines. Okay, so that's basically it. Just a quick recap: mucosal immunity, a very important component of overall immunity as well, but focus more on that interface between the external environment. And so they're regulated mainly by the innate cells, the innate barriers, and as we talked about, secretory IgA, very important part of it. So a lot of infections actually occur to mucosal routes, but we don't have a lot of mucosal vaccines available. They're a bit more complicated to formulate because they're considerations of the different routes and how the different routes and the different cells present in those mucosal tissues will affect how the vaccine is processed. So there are new technologies being looked into to improve the antigen formulations as well as the delivery of these vaccines so that they can actually reach their target. All right, so that's about it. References here, I'll put the slides up and I will inform you about the test coming at the end of this week. And then in the next week, what I'm planning to do is actually maybe go through some of the test questions or rather go through a recap of the lectures and then start preparing you for the open book exams that will come at the end. Before that, we'll have another test that just combines everything else, as in part 1 and part 2 in one test. And then we'll have an open book exam. And I'll talk more about this later on, but what you need to bear in mind is what you're covering and what you're being tested on in the tests will depend on the lecture notes. But for the open book exam, it will require much more. There will need to be some active reading, and when you want to answer the questions, you need to be able to basically do very quick literature reviews to be able to answer some of the questions. So they're not going to be questions that you can just look at your notes and be able to answer. That's why they're open book exams. So I think because it's a new thing, I don't know if any of you have ever done an open book exam before, I'll dedicate one session and possibly will allow that session to be where you can drop in and put some questions as well. So maybe I'll look into YouTube Live or something like that so that you can ask questions as I go along with a few practice questions for you to see how it works. Maybe even some past year questions just to see how to understand a question as well. All right, so that's all from me for this week. Good luck in your test, and you can obviously email me if you have anything that's not clear, any questions. All right, have a good week.